Well, hello everyone, and welcome to Island's Investor Briefing today. My name is Jane Lowe, and I'll be your host for today's session. Joining me for this session is Paul McLeeman, who's Island's Executive Chair, Dr. David Foster, Island's CEO and Managing Director, and Chris Timonopoulos, who has recently joined us as Non-Executive Director. Good morning or good evening to all of you.
Good morning.
Evening. The format for today is that the team will run us through some of the presentation slides that were lodged to the ASX this morning, and we'll discuss news last week of a placement and that the phase 2A cohort in the phase 2A/2B clinical trial, the PROTECT trial for ILA 101, had all been dosed. After that, we'll open up the floor to Q&A. If you'd like to ask a question, you can do so using the Q&A ribbon in the bottom of your screen. But with that, I'll now pass the floor over to Dr. David Foster, who will get us on the road. Thank you, David.
Very good, Jane. Thank you very much. Thank you all for joining. I'm very excited to give you an update, tell you what's gone on with the company. Many of you know the history and what's gone on up to this date. As Jane alluded to, last week we had a couple of really exciting announcements, and I'll get you up to speed on that. So just to set the stage and make sure we're all on the same page here, Island Pharmaceuticals is an antiviral drug development company. We are a clinical stage drug development company, and in fact, a phase 2 clinical company. So we're excited about that. We're focused on infectious diseases. The assets that we look at that we think are important are molecules that would come to us with known small molecules with clinical history. We like mid to late stage clinical or other abbreviated routes to approval. We think that is a very key component of our strategy. We like to look at molecules that have open INDs when we see them, and where we may have validated government or military funding, non-dilutive funding opportunities. So these are the things we look at, as well as priority review vouchers. The molecules we'll tell you about today have all of these features. Island at a glance. So we have a lead molecule. It's called ISLA 101. ISLA 101 is a molecule. It's got a well-known history. It's been around for many, many years. Well-known safety profile. It was initially developed by a large pharmaceutical company as a cancer drug, yet it's never been approved for anything, primarily because it hasn't been demonstrated to be an effective cancer drug. However, we have data that I'll tell you about demonstrating that it is very good at being an antiviral molecule. So the type of viruses it targets are mosquito-borne viruses. So think dengue, West Nile, Zika, yellow fever. ISLA 101 has activity against all of these viruses, making it a unique pan-antiflaviviral molecule. There's 400 million infections of dengue every year. That's actually more than malaria. It's a huge unmet medical need. There's no therapeutics. There's no small molecule prophylactics. There are a couple of vaccines that are coming onto the market. They are imperfect, to say the least. There's a lot of room for improvement on the vaccine front. So we think that creates a huge opportunity for Island and for ISLA 101 with major market potential. Again, 400 million infections around the globe. The preclinical data package that I'll show you in more detail as we go through the presentation is robust. It comes from world-class labs, one at Monash University, the other from Harvard. These two labs were publishing results at nearly the identical time. So we feel great that our molecule has been studied by these phenomenal institutions, and they each validated the other's work, which puts us in a very unique situation. I'm very happy to let you know that we dosed the phase 2A cohort of the PROTECT study last week. That study is off and running, and we're excited to see the results in the not too distant future. One of the other unique opportunities about ISLA 101 and galidesivir, the other molecule we're looking at, is that they're eligible for priority review vouchers. Many of you may have heard me talk about what I'm going to call PRVs. These are an incentive created by the FDA to really encourage companies to develop drugs for underserved diseases. As it turns out, dengue, chikungunya, Zika are on the list of diseases for which you can get a PRV, as well as Ebola and Marburg and some of these other really nasty viruses. Once you get your drug approved, the way the PRV works is you get a voucher that enables you to get fast track approval of another drug following new drug application submission, and you can get expedited review at the FDA. These vouchers are sellable. They typically sell for about $100 million US. So ISLA 101, our existing program, is currently eligible. Galidesivir, the other program we're looking at, also is eligible. And as I mentioned here, pipeline expansion, we are actively looking at other molecules. As you may have seen, we've announced that we have a letter of intent, binding letter of intent for an option to acquire the galidesivir program, and we're actively working on diligence right now. So many of you have seen this slide. This is a slide that we update nearly every time we give a presentation because it shows dengue outbreaks from around the globe over the last 3 months. So this slide changes nearly every time we give this presentation. And to me, it is one of the most frightening descriptions of what we're seeing with dengue all over the world. It's no surprise, you can see for yourself, South America, Central America, they are hotspots. They're overrun with dengue, as is India, as is Southeast Asia. Huge problems there. But what we've seen, even since we started the company, is we're seeing the mosquitoes that carry dengue, and therefore the viruses, moving into places they've not been seen before. So moving away from the equator, both north and south. And now we're finding in the southern part of the United States, Los Angeles in the United States has a locally acquired dengue outbreak. San Diego today announced the first locally acquired dengue outbreak or dengue case. So it's a huge problem and it's only growing worse. And in fact, it is predicted that by the year 2050, these regions that are highlighted in yellow and red and orange will be endemic for dengue. Now, look at the northern third of Australia. It's predicted to be a huge problem here. The southeastern third of the United States, as well as the southern part of Europe. And in fact, we're seeing dengue moving into Europe right now. Different mosquitoes are carrying dengue. It's a growing problem. And again, there's really nothing you can do about it right now. Again, to ensure that we're all on the same page, I've talked a lot about mosquitoes. The way that dengue is spread is through the bite of an infected mosquito. So you can be uninfected, but somebody who lives in your house may have dengue. If a mosquito bites that person and then bites you, the mosquito will be the vector that transfers that infection to you. And the cycle can continue and go round and round. So while you don't get it by breathing on each other, you do get it when there are mosquitoes around, which is why there's such a huge effort for mosquito control. In certain parts of Southeast Asia, Singapore, for instance, it's very, very strict to get rid of water, to eliminate standing water and try to reduce the level of mosquitoes. There's a variety of mosquito eradication efforts, but they're not going to work by themselves. We're going to have small molecules, likely have to have vaccines as well if we're going to cure or curb the problem associated with the dengue infection. The World Health Organization reminds us that half the world's population is now at risk of infection. There's more than 400 million infections with dengue every single year. So let me tell you about ISLA 101. ISLA 101, as I said, was initially developed by J&J. It was initially developed as a cancer drug. It's been in 40 plus clinical trials. It's been in thousands of people in the clinic, not including our own clinical trials, but it's never been approved for anything. And again, not because for safety reasons, but it's because it hasn't been demonstrated to have robust anti-cancer activity. When we began looking for a molecule that had activity against these mosquito-borne viruses, we found publications coming out of Monash University describing that ISLA 101 had been identified from a library of molecules, looking for molecules that had anti-dengue activity, in short. And in fact, it's been demonstrated that ISLA 101 has activity against all 4 strains of dengue, as well as West Nile, Zika, and yellow fever. Soon after that work was published, a group at Harvard published nearly identical results. They performed a screen, they isolated ISLA 101, demonstrated preclinical and animal activity in mouse models. So the data package is robust. The labs that have looked at this are fantastic, world-class labs. And on top of that, we have 40 human clinical trials worth of data demonstrating that this molecule has a well-known safety profile. So we are excited to have had the chance to move the molecule ISLA 101 into the clinic. We completed a phase 1 dose escalation study earlier this year that was conducted in Australia. The nice thing about that study is it gave us very, very good idea about what dose we need to use in an upcoming phase 2 clinical trial, the PROTECT clinical trial. So we're moving forward with the PROTECT study now. David Jans and his team at Monash University did a really elegant job of studying ISLA 101. They wanted to find out the mechanism of action. How does ISLA 101 work? And in short, I'll tell you, when dengue, the virus infects a human cell. It gets into the cell, and then it really turns the host cell into a virus making machine. And so the virus is turned into these viral proteins. One of the viral proteins needs to get into the nucleus of the host cell. This is the NS5 protein. And if I had a pointer, I would point to it here. But NS5 has to cycle in and out of the nucleus. And it was determined by David Jans and his group that ESL 101 actually prevents the nuclear uptake of NS5. So if NS5 can't get into the nucleus, the virus doesn't propagate normally. So that's that mechanism of action. The other really important piece of work that David Jans did is shown here on the right. In this experiment, he was looking to see what's the concentration of ESL 101 that is effective at preventing the virus. Straightforward question. So what we're looking at along the x-axis is an increasing concentration of ESL 101, and along the y-axis, we're looking at virus level. And what you can see, there's a term here, EC50. This is the concentration at which the virus is 50% of the virus is inhibited. And that concentration here is 0.81 micromolar. And that's a really important number, because we know from our phase 1 studies that we can achieve blood concentrations of ESL 101 well above 0.81 micromolar. And in fact, that's the dosing that we're going to be using in our study, or that we are using in our study right now. The final piece of data I want to show you from the preclinical work is this one, and I'm sure many of you have seen it, but it's the experiment that, to me, convinced me that I wanted to be involved with this. This is a survival curve that we're looking at. This is a mouse model experiment, and the mice are being infected with a lethal strain of the dengue virus. So what we're following on the x-axis is time, and on the y-axis is % survival. So at the beginning, all of the animals are alive. By day 5, the animals that received no drug were all dead. This is the red line that you see on your slide. However, if you give these animals 1 dose a day of ESL 101, about 20% of the animals survive. If you give them 2 doses a day of ESL 101, about 70% of the animals survive. Now, one of the things we know based on the work that out of our phase 1 study is the dose that we plan to use and that we are using in our phase 2 study is, when calculated to look at the differences between mice and humans, this is about, we're going to be dosing at about 10 times the level that was dosed in this experiment. So well above that, which has already been demonstrated to be effective. So as we move into what we now call the PROTECT study, this is a prophylactic and treatment challenge trial. And you've heard me talk about challenge studies. In a challenge study, you enroll healthy individuals, and then you infect them with a weakened or an attenuated strain of the dengue virus. It was a virus that was initially developed to be a vaccine, but it was just a little hot. It made people a little uncomfortable, a little bit sick. So the Army determined that, well, we could use this and study this virus in humans and learn a lot about a dengue infection, and that's exactly what they've done. So the Army has run phase 1 clinical trials looking just at an infection with this virus, the same virus that we're using in our study, and asked what happens to these folks. And I can tell you, the individuals who get this, they get a little pain, they feel a little achy, some run a fever, most get a rash, but they get clinical dengue, but it's a very mild form of it. So I mentioned that the Army's run a phase 1 study. We actually have a CRADA, Cooperative Research and Development Agreement, in place with the Army, under which they're giving us all of the data out of that phase 1 study, and they're giving us the virus that we can use in our study. And in exchange, they've asked to see our data. So we're more than happy to share with the U.S. Army the results from our study. If we look on the right here, the study will be conducted, is being conducted at SUNY Upstate in Syracuse, New York. We're running 2 different cohorts. We're going to be running, the first cohort is a prophylactic study. The second is a therapeutic study. So we're going to look at the ability of ESL 101 to either block an infection or to treat an infection. And let me show you what I mean. Well, I'll come back to this on this slide. On the left, you're going to see the prophylactic study. In this study, this is what was dosed last week. We dose individuals with ESL 101. Then 3 days later, we'll infect them with the attenuated version of the virus, attenuated dengue virus. And then we follow them. These individuals come to the hospital every day or every other day, and we take blood samples, and we can measure all kinds of stuff, not the least of which is the virus level. We can measure the fever from these folks. We can measure a variety of blood cell counts. So much information. When we finish this study, which we expect to have data by the end of this calendar year, we'll then look to the second part, the phase 2B arm of this study, which is a therapeutic arm. And in this arm, we will initially infect individuals, and then by about day 7, we will dose them with ISLA 101. Here, we expect these folks to be gently viremic. We expect some symptoms to be appearing, and we want to see if we can curtail the infection and reduce the symptoms in these individuals. I wanted to go back to this. Stephen Thomas, who's on our scientific advisory board, also is head of infectious diseases at SUNY Upstate, where we're running our clinical trial. And you may have seen, we recently did an interview with him. He's super helpful. But we asked him to describe what are the benefits of a challenge study? Why would people do a challenge study? And Stephen very correctly said, when there are no available animal models of a disease, researchers take a weakened form of the virus and create a mild, safe form of the disease in a healthy person. The idea is that in a safe, well-controlled environment, using a small number of people, we can get information that normally would involve going into the field and exposing a lot more people. That's one of the benefits of doing this that we've touted for years, literally. Stephen continues, it is good timing for Island to test their product, ISLA 101, now that it has successfully passed a phase 1 safety study. The phase 2 trial can then examine in a small, very controlled and safe environment if ISLA 101 has an impact on viral replication in humans, the symptoms, and the immune response. So again, it's spot on with what we've been saying and why I'm so excited about this study. This is a lot on this slide, and I know that, but I think it's important because what I'd like to do is point out some data that we can get from the challenge study. So this is data from the study that the Army ran that I've already told you about. This is results from one individual, and I think it just highlights how really powerful this study is going to be. So in the first instance, if you'll follow the blue line, this is virus level. In one individual, this is the virus level over time. And you can see that by day 4, the virus level starts to increase. It peaks at about day 10, and then the virus level is actually gone by about day 14. The individuals continue to come into the hospital. They continue to get their blood drawn because we want to follow more things. For instance, we're interested in studying the antibody response to the virus. This is shown in the lines with the triangle and the circle, the green circle. You can see there is an increase in the antibody response to the virus. In the red line here, we can look at the fever that this person had. And in fact, this particular individual didn't have much fever. Some of the other ones, they do have fever. Fever varies in this particular model. But this individual did complain about abdominal pain, eye pain, fever, nausea and vomiting, as well as a rash. So these are all classic dengue symptoms. And what we would, of course, hope is that we can suppress viremia. We can reduce these symptoms. And again, out of the prophylactic arm, we'll know this by the end of this calendar year. And by the therapeutic arm, which will start in January of 2025, we expect to know those results by the end of Q1, early Q2 next year. So as we look to other molecules, and I've already mentioned this at the beginning, some of the things we look at are, does the molecule have clinical history? Is it a small molecule with antiviral activity? Is it eligible for a priority review voucher? Might the military be interested? Might there be sources of non-dilutive money? And is it a fit for our management team? We have developed connections and brought to the company connections and relationships with different militaries, really in different countries. And so we're hoping to be able to capitalize on that. I want to switch now and tell you where we stand with galidesivir. Galidesivir is a molecule that we've discussed recently in some of our announcements. BioCryst, a company in the United States, has been developing galidesivir, and it's a very, very interesting molecule. It is a very broad-acting antiviral. It's been in development for a number of years. It's been supported by tens of millions of dollars of US government money, which we find very attractive. Some of the viruses that galidesivir has activity against would include Ebola, Marburg, Zika, many other RNA viruses. These are viruses for which there's really no treatment. There are significant problems and at huge risk around the globe. So we've signed a binding letter of intent for an option to acquire galidesivir. We are performing our ongoing due diligence right now. We're deep into that right now. Some of the features of galidesivir that we like is that it comes to us with substantial phase 1 human safety data. It also comes to us with multiple non-human primate studies with different viruses, such as Ebola and Marburg. Very rigorous experiments, very tough experiments to do, but their data package is fantastic. There may be an opportunity to pursue an expedited path to approval using the FDA's animal rule, which the animal rule says if it is impossible or unethical to conduct an efficacy study in humans, you may be able to substitute studies in animals rather than the human study. We're exploring that possibility right now. And again, it would be eligible for priority review vouchers. The commercial opportunities with both of these molecules are really largely overlapping. We think on the one hand, they may be effective for travelers traveling to endemic countries. We think the military may be interested in both of these products. And we think that both of these products could be useful for national outbreaks. In particular, I could see South America, Central America, India being very interested in ISLA 101 for outbreaks, and then around the globe, really, for outbreaks against Ebola, for instance, with galidesivir. We think the government stockpile program might be useful for both of these. And then again, both of the programs are eligible for priority review vouchers as of today. The other announcement that we put out last week, and I'm incredibly excited to tell you about it, is we announced that we have secured $3.5 million in new funding. The funding will go to support the rest of the PROTECT clinical trial, the phase 2 study that we're currently running, as well as continue diligence on galidesivir, and it actually provides enough money for us to acquire the galidesivir asset should we elect to do so in the future. The structure of that financing is it's a 2-tranche placement at 7 cents a share. There's one new option attaching for each share issued, also priced at 7 cents. 50% of those options expire within 1 year, 50% in 2 years. We are incredibly grateful to have been supported by a phenomenally strategic group of investors. This would include Dr. Daniel Tillett, Angus Walker from Hong Kong, as well as Dr. Bill Garner and Jason Carroll, investors who have supported us, and we're excited that they're going to continue to support us. I'm also really happy to see that our new director, Chris Tumanopoulos, was excited to participate and get involved with the company at this level. So we are in a very strong position right now to move forward with all of our objectives, to execute on the milestones that we have in front of us, and we have money in the bank to do so. So we're incredibly excited about that opportunity. Our corporate snapshot, I won't go through all of this in great detail. Last week, our share price closed at 13.5 cents, which is significantly up from where it's been, giving us a market cap of 16.35 million. The shares on issue, which do include the new issued shares, is 154 million. And then many of you may know that we have some listed options, 32 million listed options with an expiration date of March of next year. Cash on hand at the end of the last fiscal year was $1.7 million. The placement announced was an additional $3.5 million. And then we have indirect support by the US government of $625,000. So we will be updating our substantial shareholders whenever we get through finalizing the transaction that we're talking about now. I'll leave you with milestones. I'm not going to give you what have we done over the last year. I want to look, what have we done last week? We dosed the phase 2 study, and I, again, couldn't be more excited about this. We secured $3.5 million in financing that is going to set us up to drive forward to get through these milestones that we have in front of us and support us into 2026. Coming up, we expect data readout from the phase 2A arm of this study by the end of this year. We will continue to do due diligence on the galidesivir program. Once we move into next year, we, as I said, plan to start the phase 2B arm of that study in January of 25, with data probably coming out late Q1, early Q2. And when we get that, we'll start talking to the FDA about an end of phase 2 meeting and thinking about what is our subsequent clinical trial going to look like. So I'm going to leave it right there. I think that's everything I have. And we're available to take questions. Again, thank you all for coming. It looks like we have a good group here. And if you have any questions, don't hesitate to reach out to me. But I know we have some Q&A right now.
Thanks, David. I'd like to first pass across to Paul McLean, Islands Chair, as I know we received some questions in advance of the webinar, and Paul's going to cover off those. So over to you, Paul.
Yeah, just just briefly, and if anyone's got any further questions, they can reach out to the company with those directly. But just addressing the structure and the timing of the placement that was made last week, the board received an incoming offer from a group of sophisticated biotech investors who had done extensive due diligence on the company and wanted to support the company. The offer was put in front of the board with a set time frame, and the board took the view that we needed to accept the offer and bring the financing in to enable the company to move forward fully funded and to reduce the risk of further financing risk going forward. The market seems to have liked the transaction. Obviously, shareholders have seen a substantial uplift in the value of their shares since the transaction was announced. I would like to reiterate what David said, that we're grateful that Chris has joined us as a director and obviously is supporting the company as well. The board really is happy with the transaction and takes the view that we've really de-risked the financing aspect of the company and enable us to move forward and meet our objectives and deliver milestones for the benefit of all shareholders. Got a question there around what happens after the phase 2. I'll let David speak to this as well, but the board and management has taken the view that we have started talking to potential partners. David and I frequently attend conferences where interested parties are present. So what happens moving forward from here is we will have an FDA meeting, get an understanding from the agency, what they would like to see for a phase 3. That'll enable us to work out what that phase 3 funding need looks like. And in parallel, having discussions with potential partner companies. The company can then make a decision as to whether we raise further funding to support the phase 3 in-house, or whether the best thing for shareholders would be to transact that asset and take a carried interest moving forward in terms of royalties and a share of the PRV, and let somebody else take on the burden of running that phase 3. So really, that's an ongoing discussion. We've got active BD happening around the fenretinide asset as we speak, and we'll continue to do that as we come through the phase 2. David, you might want to add to that.
Yeah, Paul, that was super. The only thing I would add to that is somebody may wonder, what would the clinical trial structure look like? And because I tout the benefits of a challenge trial all the time. I think it's unlikely that the next study is a challenge study. I think we will be in an endemic country doing probably a therapeutic study in an endemic country, but that really remains to be seen. We need to get some feedback from the FDA on that, but I don't think it's going to be a challenge study. But otherwise, Paul, I think you're exactly right about the optionality that we want to maintain going forward.
We have another question, and I'll just read verbatim because I'm not exactly sure what it is, so we might address both possible reasons for asking the question. So it's a question's asking, what's the shelf life of ISLA 101? I guess, David, I might throw that to you, both in terms of drug on hand and also IP, because I guess it could be either.
Yeah, so I think this is a question about shelf life of the drug on hand. It's at least well over 2 years. The batch that we had made recently is undergoing ongoing stability studies right now. It's been fine up to date. The previous versions of this molecule are stable much longer than that, but I'm just telling you what I know about our existing molecule or our existing formulation and the drug that we had made.
Great. Thanks. We had a couple of others here. So, David, you announced last week that you dosed the clinical trial. Can you give us any more insight into when we might expect to see the results?
Yeah, so I showed you the data slide, right? The wild data slide. That gives you an idea of how quickly some people may be able to have an indication of how this drug is working, right? The symptoms, the virus is up and gone by about day 14. Symptoms last until maybe around the first month or so. So the individuals will all be examined during that period of time. And then thereafter, as we're taking blood samples and doing the analysis on those blood samples, we want to understand the pharmacokinetic properties of how that was dosed. I say all of that to say that we're going to get a lot of information in just a couple of months. Again, we'll have a good idea well before the end of this year how this is working and be in a position to discuss moving on from this study to the next study, the therapeutic cohort.
Great. Sorry, I'm on mute. Okay, so another one here just on a competitive product. So I've heard that J&J has discontinued the work they were doing on their dengue molecule. Where does that leave you in the competitive landscape?
Yeah, so frankly, it leaves us in a very positive competitive landscape. We have been aware of the J&J molecule, obviously, for quite some time. We knew it. In fact, we know those folks. And, you know, it sort of hurts me a little bit to see that it's not being developed anymore, but it does create an opportunity for us that I think we're now one of 2 remaining small molecules in the clinic for dengue, which presents a huge opportunity for us, to be real honest. And we're the only pan-antiflaviviral molecule that's in the clinic, right? The other molecules, including the J&J molecule, are very specific for dengue. That's not us. We have activity against a variety of these flaviviruses. So we're in a really strong competitive position right now.
Great. So another one here, just on the PRVs, how much of a market is there for them at the moment? And any idea of the recent sale averages?
Yeah, that's a super question. Over the last 2 years, PRVs typically have sold for about an average of $105 million US. There's a couple of points I want to make on PRVs. But the short answer to this is the most recent one sold for $150 million US. Huge uplift. And I'll be very interested to see what happens to the next one. The next one, there's a couple of flavors of priority review vouchers. We're pursuing a tropical disease PRV. There's also a pediatric PRV. The pediatric PRV was set to expire in the United States. It just got extended. I think yesterday or today, it got extended only until December of this year. So Congress in the United States is, I think, working hard to try to get that extended. Nobody wants to see it go away. But the good news for the tropical disease priority review voucher program, it doesn't have a sunset on it. So we're going to continue to be in good shape. But the average PRV, about 105, the most recent one, 150.
Amazing. Okay. So on galidesivir, could you give us an update on what you see as being the next sort of steps in the due diligence process as being?
Right. So we have done, and Paul's done a great job assembling a team of individuals, subject matter experts, looking at a whole slew of things. We're looking at, obviously, IP, the CMC, preclinical work, interactions with the FDA, and there's a lot because the molecule's been around for a long time. So it is a big piece of work. But we're very happy to be doing it because it's a very exciting molecule. So what you'll see over the coming, maybe by the end of the year, we do have definitive documents that we need to finalize and get executed. We're working towards that right now. And then after that, we'll have a continued option period until we decide to exercise that option and acquire the asset.
And can you just talk a bit more about the DoD funding that was applied to the ELA 101 study, which was announced a while back?
A while back. Yeah, this was so incredibly helpful for us, Jane. The Department of Defense had given SUNY Upstate a large grant to conduct research on the samples that came out of our clinical trial. And the funds were earmarked for studies that were not included in our trial. So things we weren't going to do, they were going to do what I call bonus science, if you will. Really important work, but we didn't necessarily need it. After talking with SUNY Upstate, they actually went back to the DoD and asked if they could reallocate some of that money to pay directly for some of the work done on our study that we otherwise were going to have to pay for. So it turned out to be, I think it was 625,000 US that we just didn't have to come up with. And I couldn't be more grateful to SUNY for asking and for the DoD for saying yes, because it really came at a great time for us and is going to enable us to, well, to execute on that plan sooner rather than later, as we've started last week.
Thanks, David. And just one related question on that. So with the CRADA, perhaps you can just explain that in more detail, how that works and the feed-in data from the Army's earlier study.
Yep, my pleasure. So I mentioned in the talk that we have a CRADA, which is Cooperative Research and Development Agreement. This is in place with the U.S. Army, and it was initially put in place so that they would give us the virus. But we decided that we asked for a little bit more. We said, we know that you've been running phase 1 studies. Those data are really important because it's the very same virus, right, that they ran these phase 1 studies in that we're using in our clinical trial. And we asked if we could use those data as comparator arms. And they said yes. Excuse me. So they wrote that into this cooperative research and development agreement in place, research and development agreement, to give us the virus, to give us the data, and then in exchange, we will give them the data. They want to see, you know, the individuals in our study, the controls who don't have drug, that goes into their database, right? So they want to see that, and they want to keep track of how everything's going. I'm sure that they're also interested in the real results. What is, do we see any efficacy with ESO 101, right? So I think that it's a two-way street, right? But it's good to be talking to the Army with a two-way street.
For sure. Sorry, Paul, go ahead.
And just adding to that, obviously the Army's potentially a big customer for this product ultimately, both in terms of deployment and stockpiling. You know, having a close relationship with the Army and an open dialogue with them really ensures that we're developing a product that, you know, amongst other stakeholders that they're going to support and like and ultimately buy. So, you know, there's the technical aspect of the collaboration, but there's also a really important commercial aspect to it as well.
Excellent. Thank you. Okay, well, we are coming up to our time for this particular webinar. I would just suggest if there are any final questions to pop them into the chat. And reminder that if we haven't covered everything today that you're sort of wondering about, that you're welcome to reach out to Investor Relations. So feel free to do that, info@islandpharmaceuticals.com. I'm conscious, I think, Paul, you sort of wrapped up the answers to a number of questions that we'd received in your opening comments at the start of the call.
Yeah, I just want to reiterate, just to be completely clear, that the board's acted in the best interest of all shareholders. We've reduced financing risk. We're now fully capitalised to achieve the milestones that we want to achieve, and we've you know, achieved a good outcome from a share price perspective. So, you know, we do act in the best interest of all shareholders. That's our remit as company directors, and that's the basis that we've moved forward with this financing.
Okay, excellent. Well, with that, David, I might hand across to you for any final sort of closing comments.
Yeah, my pleasure. One, Chris, Jane, and Paul, thank you for being on and for coordinating this, Jane. For those of you who attended, thank you very much. We appreciate your support. I hope you're excited to be along with this next part of the journey with us. It's going to be a phenomenally exciting next 6 to 9 months as we execute on a clinical plan, as we look to bring in a new molecule. It should be a phenomenally fun 6 to 9 months. So I think that's all I've got.
Excellent. Okay, well, with that, I'll thank everyone for joining, and we look forward to keeping you across Island's progress. Thanks, everyone.